My name's Keith Jung. I'm from Mass General Hospital, and I had the great pleasure of being able to host the panel today entitled Gene Editing, Achieving Therapeutic Mainstream. Before we get started, I wanted to provide a little bit of background to the topic. As I'm sure many of you know, gene editing is an exciting set of technology platforms that enable targeted alteration of gene sequences with high efficiency and the ability to do so in living cells or organisms. As you might expect, there's tremendous excitement for these technologies, not only for research applications, but also for the development of novel therapeutics and for a wide range of different human diseases, including many for which there are no effective treatment options. So I'm really grateful today to be able to welcome our panel members who are all leaders in some of the biotechnology companies seeking to move gene editing technology to the clinic. Today we have on our panel John Evans, the CEO of Beam Therapeutics, Rachel Horowitz, the president and CEO of Caribou Biosciences, and Lisa Michaels, the EVP and Chief Medical Officer of Editas Medicine. So welcome to you all, and thanks for joining us for the panel today. I thought I would start with a question that would give you the chance, each of you, the chance to be able to both introduce yourselves as well as your companies. And so the two questions I want to pose to you are, what technologies is your company deploying specifically? And what are some of the indications you're pursuing? So why don't we go ahead and start with John?
Sure, thanks, Keith. And it's great to be here, and thanks everybody for the interest. I'm John Evans, CEO of Beam Therapeutics. Beam is in the CRISPR field, so we're all part of the gene editing field, which is obviously moving very quickly. We use a newer form of CRISPR, which is called base editing. Base editing basically uses CRISPR for targeting the genome, which is an amazing feature of CRISPR. It's very easily programmable. It's very flexible to sort of target different parts of the genome with very high specificity. In our case, when we get to that site, instead of what is traditionally done, which is making a cut or a break at the DNA, we don't do that, so we turn that feature off. And instead, we sort of open up the DNA, and then instead, another domain is added to it, which can basically create a single base change at the target site. So now what we're doing is effectively making single letter changes in the genome at targeted sites. So why is this interesting? So it turns out that over half of all genetic changes in disease are point mutations or single letter changes. These are things like sickle cell anemia, alpha-1 antitrypsin deficiency. And so we would be very interested, of course, in landing an editor right at that spot, finding that one letter that is misspelled in the gene and causing the disease, and turning it back to something that's normal. And so those are indeed two programs that we have ongoing at Beam. It turns out we can do many other things with that system as well. So we also can use single base changes to turn off genes, so we can silence genes, we can activate genes, we can even reprogram protein function. So you sort of change a single letter, you change an amino acid on the surface of a protein, you change how it will function. So base editing has been a very interesting and broadly versatile genome modification tool. And so at Beam, we're really designed to try to bring this broadly to patients wherever they can benefit. And so we're doing work in hematology, with sickle being our lead program, immunology, of course, creating CAR T cells with high levels of engineering, and then in vivo editing with lipid nanoparticles to the liver, things like alpha-1 antitrypsin deficiency, as I said, and then AAV viral vectors to the eye as well. So very broad portfolio of different delivery modalities to bring this newer editing technology to patients wherever genetic medicine can be practiced.
Okay, thanks, John. Rachel, why don't we turn to you next, what you're doing at Caribou?
Sure, thank you, Keith, and thank you for the invitation to participate today. So Caribou is a clinical stage CRISPR company spun out of Jennifer Doudna's lab at UC Berkeley. And today we're using a CRISPR technology that we call the Chardonnay Technology. And Chardonnay stands for CRISPR Hybrid RNA DNA, and it describes the new guides that some of my colleagues developed here at Caribou. Rather than relying on the all RNA molecules that typically target a CRISPR protein to the right site in the genome, they've instead developed hybrid molecules that contain both DNA as well as RNA. And we call these the Chardonnay guides. We use these because they significantly improve the specificity of genome editing. And we think that's really quite important as we look to bring to bear ever more sophistication to the genome editing that we can do to cells for therapeutic purposes. Our initial focus at Caribou is to use this technology in order to develop allogeneic cell therapies with best-in-class potential for both hematologic malignancies and solid tumors. In the hematologic setting, we have programs for non-Hodgkin lymphoma, multiple myeloma, as well as AML. These are all allogeneic CAR Ts. And our allo CAR T for NHL is in a phase 1 clinical study now. Additionally, we've developed the capabilities to genome edit iPSCs, induced pluripotent stem cells, and differentiate them into natural killer cells. And we use these in order to develop CAR-NKs with a specific focus on solid tumor malignancies. Of course, we see this really as the tip of the iceberg. As you've already heard from John and you'll hear from Lisa, there are many ways that CRISPR technology can be deployed. So we're excited about these initial applications.
Great, thanks, Rachel. And Lisa, over to you and what you're doing at Editas.
So Keith, and so I'm just to introduce myself again, Lisa Michaels, and I am the Chief Medical Officer at Editas. Way of background, before I joined the industry about 15 years ago, I was also for almost 15 years a pediatric hematologist oncologist who was really challenged by having to treat children who had diseases that just honestly had no alternative treatments available to them. And the beautiful part about moving into the editing technologies is the ability to actually target in a very specific manner, making changes that can actually treat diseases that are not only inherited, but are also acquired due to the problems related to genetic abnormalities. So it was very exciting for me to be able to join Editas about 6 months ago, where we have a very broad portfolio in which we're moving forward, not only in looking for cures for congenital causes of blindness and loss of vision due to retinitis pigmentosa, but also ex vivo cell editing, as specifically targeting thalassemia and sickle cell disease at this point in time, and also an increasing broadening pipeline, which allows us to target iPSC, specifically looking at potential treatments for solid tumors. A little bit of overlap from what you've heard from both Jim and Rachel. Again, it's a CRISPR editing company, but I think the thing that is very nice is to be able to show that just because it's CRISPR editing doesn't mean it's one size fits all, and that every one of these particular techniques has the ability to treat the same diseases. In fact, each one of them has a certain advantages and disadvantages, which basically says we can all exist in the same place targeting those diseases. I think the challenge is, as good as we're getting at editing the genome, the real challenge is trying to actually deliver the technology into the cells that need to be treated. And that's one of the things that's a strength right now with Editas, is the fact that we're playing both in vivo with AAV-based delivery, as well as ex vivo editing, and plus also being able to be the cell lines. I think the one thing that I would just point out is that a lot of our technology is actually based on the Cas12a enzyme, which has distinct advantages related to the size and the efficiency of being able to deliver, which we also think will be able to provide a best-in-class and also be able to target different diseases than some of the other editing platforms.
Great, thanks. Actually, you just brought up delivery, Lisa, so why don't we talk about that next? So as you said, we've gotten very good now at being able to target changes wherever we want them in the genome. But now the challenge, one of the challenges going forward, is how do we actually deliver these things to cells? So maybe if I could get each of you to talk in a little more detail about what your efforts are here and what you see the challenges as with respect to delivery going forward for each of your different platforms and indications. And John, maybe I'll turn it back over to you to start.
Yeah, I completely agree with the point that I think delivery remains probably the biggest challenge in our field. The genome modification technologies are leaping forward in all sorts of interesting ways, but we still want to treat a patient at the end of the day. And that patient has a problem in some organ or system of their body or their whole body, and we have to get it into every cell that matters, right? And so there are some tried and true methods at this point in genetic medicine, as I know over the course of this conference has been discussed, whether it be ex vivo, taking the cells out of the body, editing them, putting that back in. That works for blood cells and T cells. Or in vivo, we certainly have the ability to go to the liver, and of course the eye, the retina. These are sort of accessible organs. But there's a lot of disease that we can't treat just with those few targets. And so we still need to do better on delivery. So there's a lot of innovation in the field. I think all of our companies are working hard on this. Beam itself has been pushing in this direction recently. We recently expanded our efforts on lipid nanoparticles to go beyond the liver. We've shown initial editing data in primates at this point in the liver, which is great, but we want to go beyond that. We want to see lipid nanoparticles deliver to other organs, whether it be the blood system or the brain or muscle, things like that. We equally think that viral vectors, AVs are of course pretty well validated, but we think there's a lot more to do there. and even beyond AAV, there may be other viruses that are interesting. So it's just a challenge the whole field needs to tackle together. And as it improves, then it will, then I think all of our companies with genome modification payloads will be well positioned to bring potentially curative medicines to even more patients.
Rachel, do you want to offer perspective on this?
Sure, absolutely. First off, I have to say I completely agree with Lisa and John. This is a challenge for the entire field. And I think part of it really boils down to what are you trying to do to the cell, and therefore what do you need to deliver to be successful at that? Certainly as we think about the work we're doing at Caribou, we're focused on off-the-shelf CAR-Ts and CAR-NKs. So we're in an ex vivo setting. We have some flexibility as we think about how to get the reagents for CRISPR into the cells of interest. And in fact, we can use electroporation to directly deliver a CRISPR protein like Cas9 or Cas12a pre-complexed with its guides. And so that's a very high efficiency way to deliver ex vivo. However, that's only a piece of the puzzle, right? Part of what we need to do is actually insert one or multiple transgenes into the T cell genome to really empower it with the appropriate functionality to be successful for patients. And so that leads to yet another layer of complexity of how you get donor DNA into these cells in order to do site-specific gene insertion. Today, we rely on AAV6, which is a version of AAV that happens to be well suited for T cells, but I would say has a number of drawbacks as we think about the long term. So we're certainly very interested in continuing to pursue other technologies for other ways to deliver large quantities of DNA for inserting new genes in this context. It's obviously only the tip of the iceberg then as you start to think about the complexity of doing site-specific gene insertions in vivo as well. And certainly this is an area where I hope there's a lot of continued activity and effort in academia to really continue to unlock new ways to deliver these kinds of payloads to different cells and organ types.
Great, thanks, Rachel. Do you care to elaborate a little bit more about, you referred to some of the challenges with AAV6, for example, maybe for those in the audience who are less familiar with those challenges, maybe elaborating a little bit more on that?
Yeah, absolutely. I think one of the big challenges is it simply requires a lot of it. And so I think, companies like Caribou and our peers are really pushing the envelope in terms of the worldwide capacity for making sufficient vector for making these kinds of cell therapies. And so I think that there are potentially some very intriguing ways to really move outside of the world of viral vectors in order to deliver appropriate quantities and qualities of DNA to different cell types.
Great, thank you. And Lisa, I know you brought up the topic originally, but do you want to add anything?
No, I think, even to the last question they asked of Rachel, I think if last year has taught us anything, it's that viruses can be very effective modalities for delivery of genetic material. At the end of the day, it's the specificity of hitting the targets, the immunogenicity and the effects of that, and our ability to actually get, as already said, enough of it to the targets that you want to get at without having immunogenic problems. So I do agree that the next stages of development in this area probably are going to move away from viral vectors. But then we also have the advantages of being able to actually specifically edit cells and change their behavior. Our particular case is that we are targeting NK cells and to be able to modify the DNA. So once you have a relatively well-established cell line that can provide you with an off-the-shelf modality, there's a fair amount of power in that mechanism too. And at the end of the day, it's really just being able to get enough of it to be able to treat the patients in a fashion that allows us with good clinical outcomes. And that's what I focus on.
Yeah, it's interesting because the gene therapy field obviously has provided a lot of knowledge and a lot of know-how to the gene editing space, but obviously the applications are a little bit different. With classical gene therapy, at least, we tend to think of things that we want to persist in the cell, whereas with the gene editor technology, it's more of a hit and run. So the challenges are definitely different. Lisa, once again, you kind of provided a segue into the next topic I wanted to talk about. You mentioned the specificity, the idea that you want to hit the target gene and not other things in the cell. There's also the broader organismic level issue of wanting to hit the right tissue and not hitting other tissues that you don't intend to hit, which obviously is related to the delivery we just talked about now. Rachel, I guess I'll start with you on this one. You had mentioned, I think in passing in your first answer about Chardonnays, and you think that they have increased specificity. So maybe if you could talk about your efforts to improve the safety and specificity of your platform and how the Chardonnays actually do what you're saying.
Yeah, absolutely. Specificity has really been a driver of our research at Caribou for years, with really the goal to do a better job of ensuring that we edit the one site that we intend to and avoid off-target consequences elsewhere. And I'd say there are really multiple reasons for why we think that's important. Obviously, first is we want to avoid accidentally editing other genes or loci that might have negative consequences in terms of the function of the cell or the long-term safety of the cell. But there's another layer, and that really relates to genomic integrity. It turns out if cells experience multiple double-strand breaks at the same time, which could be both an on-target edit and an off-target edit, one of the ways cells can repair those breaks is by undergoing massive chromosomal translocations. And so we also, of course, want to diminish the risk of those from occurring in our cellular therapies as well. So obviously improving specificity is something that you and many others have been working on in the field for quite some time. And I think our team had a slightly different viewpoint from many of the folks in the field. Many of us are RNA biochemists by training, and so we look at a CRISPR system like Cas9 or Cas12a, both of which we use. We see a lump of protein that's interesting, but maybe gets in the way sometimes, and a fascinating piece of nucleic acid that could be used for engineering. And so we really centered our work on the guides themselves and found that by including DNA in them, we actually decrease the affinity of the CRISPR complex for its target. And that helps us really reduce the frequency with which we see any off-target events. We're able to combine that with a proprietary delivery technology that we can use ex vivo that allows us to actually carry out multiple edits in a population of cells and reduce the risk of those translocations from occurring between the sites that we're targeting. So that really unlocks the opportunity for us to carry out multiple edits with high specificity and high genomic integrity.
Yeah, that is a challenge, I guess, when you're doing things like CAR T, where you want to make multiple edits simultaneously. Lisa, I'll turn over to you next. Do you want to talk about this topic a little bit and Editas's efforts in this regard?
I come from it again from the clinical perspective and not the highly detailed scientific perspective that Rachel comes to it. But at least, I'm very content related looking to a lot of our non-clinical data regarding the actual affinity and the ability and the specificity of being able to make the edits in exactly the targets. I think the other thing is also is that at the end of the day, it's really going to be the long-term safety that's going to be what we're going to observe over time. I know there's been a number of concerns recently in the gene therapy region where patients have either just by chance or due to potential concerns related to integration have actually had some cancers that have raised concern about the safety in this particular space. And I think that's one of the potential drawbacks actually of the gene therapy approach as opposed to the gene editing approach, where foreign DNA is sitting in the cell and potentially could be incorporated when breaks take place at any point in time due to whatever challenges take place in the cell division. So I think to a certain extent is that if we're able to actually be able to have higher efficiency of editing, there's a potential that we're actually going to bring in a safer type of modality as long as we're able to assure that we're hitting exactly the targets that we need and want.
Great. And John, I'm going to give you the last word on this topic.
Sure, and I'm going to riff a little bit as well on what was just raised too. I think off-target through your work and others has really advanced dramatically. I think our ability to measure off targets to very low degree is incredible at this point. And I think if you do your job right, you can create products that have minimal to no observable off target. So I think gene editing is arriving on that front, and obviously it's critical. So I think the control of off target is probably a little farther along than people guess, and I think that'll become clear as the field goes forward. And it needs to be. These are going to be permanent changes. With Beam also, I'd just say, base editing, we have this advantage of not cutting the DNA. So when we do cell therapy, we can also avoid those translocations that Rachel mentioned in terms of just not making the breaks in the first place. And that's been an advantage. So we can make cells that have many different edits. We have a cell product going into a pediatric leukemia with about 4 edits each. So that's an exciting application. So I think in general, this is all moving forward. The one thing I wanted to riff on was the idea that comparing us to gene therapy, I think, is quite interesting. We were talking about delivery before, and I think it's important to realize delivery is hard for everybody, but the gene editing with this sort of hit and run context, you really have a lower bar in delivery. I mean, we just need to get the editor in once for just enough to make the edit, and then the gene takes over the rest of the therapeutic effect, whereas the gene therapy needs to create a therapeutic level of a transgene forever. And so I think in many cases we have a slightly lower bar for delivery. And then I agree at the point that on safety, I think there is a decent chance that gene editing turns out to be safer just because we have more control, and then the machinery is gone once you've made the change to the genome. And so as long as you have control of those changes, I think it has every chance to be, a really clean outcome for patients long term.
So I think we have time for one more topic. And so I thought I would jump to this idea about patient uptake and acceptance. We've seen, for example, with the coronavirus vaccine, that there are varying degrees of hesitancy around having things that are at least perceived to be genetically oriented. And wondered if you wanted to talk about the way you think about this and the way your company's thinking about the challenges potentially of how to get patients to accept and uptake the therapies that you're developing. I guess here, Lisa, I'll start with you.
No, I think it's fun actually watching the concerns people have had related to the vaccines, where they hear DNA and RNA and they get a little worried and concerned. And I think some with the hyperbole about how you're going to turn into a monkey and it's going to edit your DNA. So I think there's definitely an uphill climb with a huge amount of the population to get acceptance for it. But I think the other advantage, and a distinct advantage for at least what we're doing, is in general, we are targeting diseases where the risk-benefit favors the benefit over the risk. Where we're either targeting diseases such as sickle cell disease, where we know that there are no good options, no good treatments, and the only so-called standard of care is either chronic transfusion or bone marrow transplant, similarly for thalassemia. And for patients who are losing their vision, at the end of the day, we are very targeted in terms of the choice of target, so that the risk benefit again is in favor of the benefit. Where it starts to get more interesting is when we start thinking to the future, because if we're dealing with cancer, if we're dealing with bone marrow diseases, if we're dealing with blindness, it's very clear to define that benefit. But when you start getting into much wider diseases where the potential targets could be treatment of diabetes or congestive heart failure or other things where there is a separate standard of care, that's where it's really going to get very interesting. And I think it's going to be dependent largely upon our ability to show good, meaningful, and lasting cures, or at least significant benefits, as well as superiority over the current treatments. And I think that day is coming. It's just a question of getting there.
And you think there's some benefit probably to, over time, as people become less wary of the therapies, perhaps because we're targeting these more severe, albeit maybe rarer diseases initially, it will help with ultimately the broader acceptance in the population.
With both treaters and patients, there's always early adopters, and I'm grateful to them for actually allowing us to bring these treatments to patients. But at the end of the day, the patients really want to know what it's going to benefit for them. And that's where having all of us working together toward those goals and generating both the data and the safety, I think that's going to make a big difference in the long term.
Yeah. John, do you want to give your take on this?
Yeah, I think it was well framed. I think, we don't have the problem of like a genetically modified food where you're going straight to the entire population right away, right? And it sort of instantly forces you to kind of grapple with these issues. We're really treating very sick people and kids and trying to make them better. And I think it's very quick to see why it's so urgent to use these tools and to try. That said, I think sort of same with my answer on the off-target, I do think these medicines are going to come faster than people think to broader and broader populations. And so we'll obviously begin in places like sickle or rare genetic disorders or cancer. But I think increasingly it'll become larger population diseases, more common diseases. I think disease prevention is potentially interesting. I think regenerative medicine is going to start to increase over time. So, and there's programs active in each of those areas already, and they just have to mature. So I think over time it'll start to address broader and broader problems. And so our job is to basically make sure that comfort and familiarity with these tools is rising alongside the growth in the application space. But I'm pretty confident. I think as our collective industry and the set of companies that are pioneering in this field, we're really working hard to make sure we do great medicine and great science, that we partner with patients, we partner with physicians at leading institutions, and we partner with regulators, frankly, and then talk to the public. And I think all of that work hopefully is laying the foundation for what could be a pretty dramatic expansion of the use of gene editing in the future.
Rachel, I guess I'm going to give you the last word on this.
Thank you. I'll wrap up by saying, personally, I'm quite optimistic on this front. And I think it's because of what Lisa described. I think as you look at the risk-benefit equation, there's a tremendous amount that these technologies and therapies can offer to patients who are in dire need of treatment. I also think we have to be respectful of the fact that this is new for so many of them, and it uses buzzwords and jargon that they're unfamiliar with. And so I think part of our work as a community is to make sure that we're providing the right information to patients when they need it. And I think there are ways we can do that as companies, and I think there are ways we can do that by partnering with other organizations who already are there with patients, providing information and answering their questions. For example, I was really excited earlier this year when we closed our Series C financing that one of the investors who participated was the Leukemia and Lymphoma Society. And they're obviously a great example of a resource who patients go to and who we can partner with to make sure that we can provide some of this information to them so that they can be answering the right questions for patients to make sure that there aren't accidental barriers to access or knowledge about these kinds of medicines.
Great. All right. Well, I want to thank all of our panelists for joining us today. I know it's you're all very busy, and I appreciate you taking the time out of your schedules, some of you from vacation, as I understand it, to join us here today. It's been a great discussion, and there will be a Q&A session afterwards. So invite you to stay for that. And we look forward to answering some additional questions from the folks that will be involved in that.